What if I told you that one of the most powerful cancer-fighting tools in the world isn't locked inside a laboratory, isn't reserved for billion-dollar treatments, and isn't something you need a prescription to access? It's already inside you. Right now, within your body, trillions of microscopic allies are working silently. They are communicating with your immune system, training it, guiding it, strengthening it. And under the right conditions, they can help your body recognize and destroy abnormal cells before they become something far more dangerous. But here's the twist. These powerful defenders don't automatically stay strong. They depend on you, on what you eat, on what you feed them every single day. Scientists are now discovering that certain gut bacteria can activate cancer-fighting T cells, reduce inflammation that fuels tumor growth, strengthen your gut barrier, and even improve how patients respond to advanced immunotherapy treatments. This isn't science fiction. It's happening at the intersection of nutrition, microbiology, and immunity. And the most exciting part? You can influence this system starting today. Not with extreme measures, not with complicated regimens, but with simple, strategic food choices that send powerful signals to your internal ecosystem. In this talk, you'll discover how specific gut bacteria help fight cancer, what foods help them thrive, and how small daily decisions can shift your body toward protection rather than vulnerability. Stay with me, because what you're about to learn could change the way you look at every meal for the rest of your life. The gut microbiome plays a central role in regulating the immune system because nearly 70% of the body's immune cells reside in and around the gastrointestinal tract. This dense immune presence exists for a reason. The gut is the primary interface between the external environment and the internal body, constantly exposed to food particles, microbes, and potential toxins. Beneficial gut bacteria act as trainers and modulators of immune function. From early life, they help educate immune cells to distinguish between harmful invaders and harmless substances, reducing the risk of inappropriate inflammatory responses. A balanced microbiome stimulates the production of regulatory T cells, which prevent excessive inflammation that can damage tissues and create conditions favorable for cancer development. At the same time, certain bacterial strains enhance the activation of cytotoxic T cells and natural killer cells, which are responsible for identifying and destroying abnormal or cancerous cells. Gut bacteria also produce bioactive compounds such as short-chain fatty acids, including butyrate, acetate, and propionate. These molecules influence gene expression in immune cells, strengthen the intestinal barrier, and reduce systemic inflammation. A strong gut barrier prevents harmful bacterial fragments and toxins from leaking into the bloodstream, which would otherwise trigger chronic immune activation. Chronic inflammation is a known driver of DNA damage, cellular mutation, and tumor progression. When microbial diversity is reduced, due to poor diet, antibiotics, or stress, the immune system can become dysregulated, either weakened in its surveillance against cancer cells or overactive in a way that promotes inflammatory damage. Maintaining a diverse and balanced microbiome supports immune precision, ensuring that defense mechanisms are strong against threats while controlled enough to avoid collateral tissue injury. Certain gut bacteria enhance the body's natural cancer-fighting T cells by influencing how these immune cells develop, activate, and function. T cells, especially cytotoxic T lymphocytes, are responsible for identifying and destroying abnormal cells, including cancer cells. For T cells to perform effectively, they must be properly primed and supported by signals from other immune components. Specific beneficial bacteria in the gut interact with immune cells in the intestinal lining and nearby lymphoid tissues, sending molecular signals that strengthen T cell readiness and responsiveness. These microbes stimulate antigen-presenting cells such as dendritic cells, which then train T cells to recognize threats more efficiently. Some bacterial strains increase the production of immune-modulating compounds that enhance T-cell infiltration into tumors. This means more immune cells are able to enter the tumor microenvironment and attack malignant cells directly. In addition, certain microbes improve the expression of molecules that make cancer cells more visible to the immune system, reducing the likelihood that tumors can hide from immune surveillance. Research has shown that individuals with a more diverse and balanced gut microbiome often have stronger T-cell activity and better immune regulation overall. Short-chain fatty acids produced by beneficial bacteria also influence T-cell metabolism. T cells require significant energy to multiply and attack cancer cells, and microbial metabolites help optimize this energy production while maintaining immune balance. Without supportive gut bacteria, T cells may become exhausted, less responsive, or improperly regulated. An imbalanced microbiome can reduce T cell diversity and impair their ability to distinguish between healthy and abnormal cells. By maintaining beneficial bacterial populations through diet and lifestyle, the body creates an internal environment that amplifies T-cell precision, strength, and persistence in targeting cancerous growths. Short-chain fatty acids, such as butyrate, acetate, and propionate, are powerful metabolic byproducts produced when beneficial gut bacteria ferment dietary fiber in the colon. Among them, butyrate is especially important in influencing cancer-related processes. These molecules serve as signaling compounds that directly interact with cells lining the colon and with immune cells throughout the body. Butyrate acts as a primary energy source for colonocytes, the cells that form the intestinal lining, helping maintain a strong and intact gut barrier. When this barrier is healthy, it prevents harmful toxins, inflammatory molecules, and pathogenic bacteria from entering the bloodstream and triggering chronic systemic inflammation, a known driver of tumor development. Beyond structural support, butyrate also regulates gene expression through epigenetic mechanisms. It inhibits enzymes known as histone deacetylases, which affects how certain genes are turned on or off. This process can activate tumor suppressor genes and promote programmed cell death, or apoptosis, in abnormal or precancerous cells. By encouraging damaged cells to self-destruct, butyrate reduces the likelihood that they will continue dividing uncontrollably. Additionally, short-chain fatty acids modulate immune function by promoting regulatory T cells that keep inflammation in check, while also supporting cytotoxic T cell responses against emerging tumor cells. These fatty acids further reduce oxidative stress by lowering the production of inflammatory cytokines and enhancing antioxidant pathways within tissues. In an environment with balanced inflammation and efficient immune surveillance, cancer cells face more resistance to growth and spread. However, when fiber intake is low and beneficial bacteria are diminished, production of short-chain fatty acids declines, weakening these protective effects. The presence of fiber-fermenting bacteria and consistent production of short-chain fatty acids create biochemical conditions that discourage tumor formation and support the body's natural defense systems. Fiber-rich foods play a critical role in supporting beneficial gut bacteria because fiber serves as their primary fuel source. Unlike simple sugars and refined carbohydrates, dietary fiber is not digested by human enzymes in the stomach or small intestine. Instead, it travels intact to the colon, where it becomes nourishment for trillions of microbes. When these bacteria ferment fiber, they multiply, diversify, and produce biologically active compounds that influence immune function, inflammation, and cellular health. This fermentation process increases populations of beneficial strains, such as those that generate short-chain fatty acids, including butyrate, which are strongly associated with anti-cancer activity. Different types of fiber feed different bacterial species, which is why dietary diversity matters. Soluble fibers found in foods like oats, legumes, apples, and flax seeds form gel-like substances that slow digestion and provide steady nourishment for microbes. Insoluble fibers from vegetables, whole grains, and leafy greens add bulk to stool and help maintain healthy bowel movement, reducing the amount of time potential carcinogens remain in contact with the intestinal lining. Resistant starches, present in foods like lentils and cooled potatoes, also function as powerful prebiotics, selectively stimulating beneficial bacteria linked to improved immune surveillance. As beneficial bacteria thrive on fiber, they crowd out harmful microbes that may promote inflammation and produce toxic byproducts. Increased fiber intake has been associated with greater microbial diversity, which strengthens the resilience of the gut ecosystem. A diverse microbiome is better equipped to regulate immune responses, maintain gut barrier integrity, and suppress chronic inflammation that contributes to tumor initiation and progression. When fiber consumption is low, beneficial bacterial populations decline, reducing protective metabolite production and weakening immune signaling pathways. Consistently feeding the microbiome with varied plant-based fiber creates a biological environment that supports cellular repair, balanced immunity, and reduced cancer risk. Fermented foods introduce live beneficial microbes into the digestive system, helping to strengthen and diversify the gut microbiome. These foods are created through controlled microbial growth and enzymatic conversion of food components, typically involving bacteria or yeast that transform sugars into acids, gases, or alcohol. During this process, naturally occurring or added microorganisms multiply and produce compounds that enhance both the nutritional value and digestibility of the food. When consumed regularly, fermented foods such as yogurt with live cultures, kefir, sauerkraut, kimchi, and miso can contribute living microbes that temporarily colonize the gut and interact with resident bacteria. These beneficial microbes influence immune activity in several ways. They stimulate immune cells in the gut-associated lymphoid tissue, encouraging balanced responses that are strong against harmful invaders, but controlled enough to avoid chronic inflammation. Some strains commonly found in fermented foods, including certain Lactobacillus and Bifidobacterium species, help strengthen the intestinal barrier by increasing mucus production and tightening junctions between intestinal cells. A stronger barrier reduces the leakage of inflammatory molecules into the bloodstream, lowering systemic inflammation that can create conditions favorable to tumor development. Fermented foods also generate bioactive compounds during the fermentation process, including organic acids, peptides, and antioxidants. These substances can inhibit the growth of harmful bacteria while promoting beneficial strains. By supporting microbial diversity, fermented foods contribute to a more resilient ecosystem in the gut, which has been associated with improved immune surveillance and enhanced responsiveness of cancer-fighting T cells. Regular intake of fermented foods may also improve metabolic signaling and reduce oxidative stress, both of which influence cellular stability. When included as part of a diet rich in plant fiber, fermented foods help create a cooperative environment where beneficial microbes thrive and produce metabolites that support the body's natural defense mechanisms. Polyphenol-rich foods support a healthy microbial balance by acting as both regulators and fuel sources for beneficial gut bacteria. Polyphenols are naturally occurring plant compounds found in foods such as berries, green tea, pomegranates, dark chocolate, extra virgin olive oil, and colorful vegetables. Although many polyphenols are not fully absorbed in the small intestine, they travel to the colon where they interact directly with gut microbes. There, bacteria metabolize these compounds into smaller bioactive molecules that can be more easily absorbed into circulation, amplifying their biological effects throughout the body. This interaction is mutually beneficial. While microbes transform polyphenols into anti-inflammatory and antioxidant metabolites, polyphenols selectively stimulate the growth of beneficial bacterial strains such as Lactobacillus and Bifidobacterium species. At the same time, they can suppress the expansion of harmful bacteria linked to inflammation and toxin production. This selective effect helps improve microbial diversity, which is a hallmark of a resilient and protective gut ecosystem. Greater diversity supports stronger immune regulation and more stable metabolic signaling, both of which influence cancer risk. Polyphenol-derived metabolites also help reduce oxidative stress by neutralizing free radicals that can damage DNA and promote mutations. In addition, they modulate inflammatory pathways by lowering pro-inflammatory cytokines and enhancing protective immune responses. Some polyphenols influence gene expression related to cell cycle regulation and apoptosis, helping prevent abnormal cell growth. By strengthening the intestinal barrier, these compounds further reduce the leakage of inflammatory molecules into the bloodstream. When polyphenol intake is low, beneficial bacteria that rely on these compounds may decline, weakening the production of protective metabolites. Consistent consumption of a variety of polyphenol-rich plant foods creates a dynamic relationship between diet and microbiome, supporting microbial populations that contribute to balanced immunity, reduced inflammation, and improved cellular defense mechanisms. A diverse microbiome improves response to cancer immunotherapy by shaping how effectively the immune system recognizes and attacks tumor cells. Immunotherapy treatments, such as immune checkpoint inhibitors, work by removing the brakes that prevent T cells from destroying cancer. However, not all patients respond equally to these therapies, and research has shown that differences in gut microbiome composition are a significant factor influencing outcomes. Individuals with greater microbial diversity tend to have stronger immune activation, better tumor infiltration by T cells, and more sustained therapeutic responses. Certain bacterial species appear to enhance the maturation and activation of dendritic cells, which are responsible for presenting tumor antigens to T cells. When dendritic cells function efficiently, they improve the immune system's ability to identify cancer cells as threats. Some beneficial microbes also increase the production of cytokines that stimulate cytotoxic T cells, enabling them to proliferate and penetrate tumor tissue more effectively. This results in a more aggressive and targeted immune attack against malignant cells. In contrast, reduced microbial diversity or dominance of inflammatory bacterial strains may blunt immune activation, leading to weaker responses to immunotherapy. Microbial metabolites, such as short-chain fatty acids, further influence how immune cells behave during treatment. These compounds support T-cell energy metabolism and reduce chronic inflammation that could otherwise exhaust immune cells. Studies have also shown that antibiotic use prior to or during immunotherapy can negatively affect treatment outcomes, likely because antibiotics reduce beneficial bacterial populations that support immune responsiveness. Fecal microbiota transplantation from patients who respond well to immunotherapy has been shown in some research settings to transfer improved responsiveness to others, underscoring the microbiome's role in shaping therapeutic success. Maintaining a varied, fiber-rich, plant-focused diet may help support microbial diversity that enhances immune precision and strengthens the effectiveness of modern cancer therapies. Chronic inflammation fuels cancer by creating a biological environment that promotes cellular damage, mutation, and uncontrolled growth. Inflammation is a natural and necessary immune response designed to protect the body from infection or injury. In its acute form, it is protective and temporary. However, when inflammation becomes chronic, persisting over months or years, it can continuously expose tissues to inflammatory chemicals such as cytokines, growth factors, and reactive oxygen species. These substances can damage DNA, interfere with normal cell repair mechanisms, and stimulate excessive cell division, all of which increase the risk of tumor formation and progression. Healthy gut bacteria play a critical role in regulating and calming inflammatory responses. Beneficial microbes help maintain balance within the immune system by promoting regulatory T cells that prevent excessive immune activation. They also produce short-chain fatty acids, including butyrate, which directly reduce the production of pro-inflammatory cytokines and strengthen the intestinal barrier. A strong gut barrier prevents bacterial toxins and inflammatory molecules from leaking into the bloodstream, a condition often referred to as increased intestinal permeability. When this barrier weakens, immune cells are repeatedly triggered, sustaining systemic inflammation that can affect distant organs. An imbalanced microbiome, known as dysbiosis, often leads to an overgrowth of harmful bacteria that produce endotoxins and other pro-inflammatory compounds. These substances activate immune pathways that remain switched on, creating a persistent inflammatory state. Over time, this environment can support tumor growth by encouraging angiogenesis, suppressing effective immune surveillance, and providing growth signals that cancer cells exploit. By supporting microbial diversity through fiber-rich and plant-based foods, the gut ecosystem shifts toward bacteria that produce anti-inflammatory metabolites. This balanced microbial activity helps regulate immune signaling, reduce oxidative stress, and maintain tissue stability, lowering the biological conditions that allow cancer to develop and spread. The gut barrier protects against toxins that can trigger cellular damage by serving as a tightly regulated interface between the external environment and the internal bloodstream. The intestinal lining is composed of a single layer of epithelial cells bound together by tight junction proteins that control what passes through. This barrier allows nutrients, vitamins, and water to be absorbed while preventing harmful substances such as pathogenic bacteria, endotoxins, and partially digested food particles from entering circulation. When functioning properly, it acts as both a physical and immunological shield, reducing unnecessary immune activation and limiting exposure to compounds that can damage DNA. Beneficial gut bacteria play an essential role in maintaining the integrity of this barrier. They stimulate mucus production, which forms a protective layer over epithelial cells, and they enhance the expression of tight junction proteins that keep intestinal cells closely sealed. In addition, microbial metabolites such as short-chain fatty acids, particularly butyrate, serve as a primary energy source for colon cells, supporting their repair and regeneration. A well-nourished intestinal lining is more resilient against injury and inflammation. When the microbiome is balanced, it prevents overgrowth of harmful bacteria that might release toxins capable of weakening the barrier. If the gut barrier becomes compromised, a condition often described as increased intestinal permeability, bacterial fragments like lipopolysaccharides can enter the bloodstream. These substances trigger systemic inflammation by activating immune cells throughout the body. Chronic exposure to inflammatory molecules increases oxidative stress and DNA damage, both of which are linked to tumor initiation and progression. Persistent immune activation can also create a tissue environment rich in growth signals that support abnormal cell survival. By maintaining microbial diversity and supporting beneficial bacteria through diet, the gut barrier remains strong, limiting toxin exposure and reducing inflammatory stress that contributes to cellular instability. Ultra-processed foods damage microbial diversity and weaken immune defense by altering the composition and function of the gut microbiome. These foods are typically high in refined sugars, unhealthy fats, artificial additives, emulsifiers, and preservatives, while being low in fiber and essential nutrients. Because beneficial gut bacteria rely heavily on dietary fiber and plant compounds for nourishment, a diet dominated by ultra-processed products deprives them of the substrates they need to thrive. As a result, populations of protective microbes decline decline, and microbial diversity decreases, reducing the resilience of the gut ecosystem. At the same time, the excess sugars and unhealthy fats found in ultra-processed foods can promote the growth of less beneficial or inflammatory bacterial strains. Some food additives, particularly certain emulsifiers, have been shown in experimental studies to disrupt the mucus layer that protects the intestinal lining. When this protective layer thins, harmful bacteria may come into closer contact with intestinal cells, triggering immune activation and low-grade chronic inflammation. Artificial sweeteners have also been associated with shifts in microbial balance that may impair metabolic regulation and glucose control. Reduced microbial diversity weakens immune precision. Beneficial bacteria help regulate inflammatory pathways and support the maturation of immune cells, including T cells, that play a role in identifying abnormal or cancerous cells. When these supportive microbes diminish, the immune system may become dysregulated, either underperforming in tumor surveillance or overreacting in ways that sustain chronic inflammation. Inflammation increases oxidative stress and DNA damage, which can contribute to cellular mutations and cancer risk. Frequent consumption of ultra-processed foods may also compromise the gut barrier by reducing production of short-chain fatty acids such as butyrate, which are critical for maintaining tight junction integrity. Without adequate butyrate, intestinal cells receive less energy for repair and maintenance, making the barrier more susceptible to leakage and systemic inflammatory responses. Over time, these dietary patterns shift the internal environment toward one that favors immune imbalance and reduced biological defense mechanisms. Daily dietary choices can rapidly shift the gut microbiome toward a cancer-protective profile because microbial populations respond quickly to changes in available nutrients. Unlike human cells, many gut bacteria reproduce within hours, meaning their relative abundance can change within days depending on what is consistently consumed. When meals are rich in fiber, polyphenols, and diverse plant compounds, beneficial bacteria that ferment these substances begin to expand. As they grow, they increase the production of short-chain fatty acids such as butyrate, acetate, and propionate, which influence inflammation, immune balance, and cellular repair mechanisms associated with cancer prevention. Dietary patterns centered on vegetables, fruits, legumes, whole grains, nuts, seeds, and fermented foods encourage microbial diversity. Each type of plant fiber feeds different bacterial strains, creating a broader and more resilient ecosystem. Greater diversity enhances the microbiome's ability to regulate immune responses, strengthen the gut barrier, and reduce chronic inflammation. These changes can occur surprisingly quickly. Studies have shown measurable shifts in microbial composition within 48 to 72 hours after altering diet. When individuals transition from highly processed foods to whole, plant-focused meals, beneficial bacteria often increase while inflammatory species decline. Conversely, diets high in refined sugars, processed fats, and low in fiber can quickly reduce microbial diversity and lower beneficial metabolite production. Because the microbiome continuously interacts with immune cells, these rapid changes influence immune signaling and inflammatory tone throughout the body. Consistent positive dietary choices reinforce beneficial microbial populations, while inconsistent patterns may limit long-term stability. Over time, sustained intake of nutrient-dense, fiber-rich foods supports an internal environment that promotes immune vigilance, balanced inflammation, efficient cellular repair, and reduced exposure to harmful metabolites, collectively contributing to a biological terrain less supportive of tumor initiation and progression. In conclusion, the science is clear. The power to strengthen your body's defense system is not distant or complicated. It begins in your gut. The trillions of bacteria living inside you are not passive passengers. They are active partners in regulating immunity, controlling inflammation, protecting your gut barrier, and even influencing how effectively your body detects and destroys cancer cells. When you nourish beneficial microbes with fiber-rich plants, fermented foods, and polyphenol-packed ingredients, they respond by producing compounds that support cellular stability and immune precision. When you neglect them with ultra-processed foods and nutrient-poor choices, that protective system weakens. No single food is a cure, and no single bacteria works alone. It is the pattern of daily choices that shapes the microbial community within you. Every meal sends instructions. Every bite either strengthens resilience or contributes to imbalance. The encouraging truth is that your microbiome can shift quickly, meaning positive change can begin immediately. By feeding the right bacteria, you help create an internal environment that is less welcoming to disease and more supportive of long-term health. Your body is designed with remarkable defense mechanisms. When you align your diet with that design, you activate systems that have been protecting humans for generations. Thank you for your time and attention. I hope this knowledge empowers you to make choices that nourish your health from the inside out. Wishing you strength, vitality, and the very best of luck on your journey toward better health.